Hello ladies and gentlemen and welcome back to C.A.R. Entertainment. I'm your host Dylan Hamilton, and today guys we're going to talk about gene sequencing as a part of our A22 biology revision videos guys. Further ado, I'm going to get straight into this because gene sequencing is just another part of our gene technology revision videos and we're almost at the end. I think this actually might be the last one we do. So we just want to get through it guys and let you know what it is. So we start off by letting you know what a genome is, and a genome is just a complete DNA sequence. Gene sequencing then, what is this process? Well, this process is working out the base sequence in an organism, the order of the bases, and if the bases and the order of the bases are along each chromosome. You do a thing called genetic mapping, which is identifying exactly where a particular gene is on a chromosome. The human genome, ours, our DNA sequence, has over 21,000 genes. It actually is over 3 billion pairs in length. 2% of our genes actually only code for polypeptides, and the remainder is just a regulatory role, which is quite insane. So DNA sequencing is faster and cheaper nowadays, and it's now possible to sequence the human genome in a few hours at a cost of just several hundred quid. So what are the benefits of performing this genome sequencing? Well, you can create phylogenetic trees where genomes of different species will be sequenced, and you can make comparisons to determine the evolutionary relatedness and generate more accurate phylogenetic trees. You can also detect genetic disease where alleles that are single genes are linked with a genetic disease. Knowing the sequence will allow gene probes to be synthesized and used to test for their presence in newborn cells, therefore you detect disease there quicker. Detecting the genetic predisposition to certain diseases. So if genes possess alleles that are genetic risk factors associated with the genetic predisposition, you will then know the base sequence that provides this, and you can provide testing for the presence of this certain disease and assessing the risk of this disease occurring. So you can also analyze the genomes of pathogens, so you can find the source of an infection you have and identify the antibiotic resistant strain of bacteria that is there. This enables scientists to track the progress of a potential epidemic. You can create 3D models of protein structure because genetic code can be used to determine the primary structure, and information on the bonding between the amino acids will determine the folding arrangements, which then allows structures to be predicted, facilitating the production of therapeutic drugs. And finally, it allows you to produce personalized medicine as it allows genetic testing, and drugs can be prescribed to match the individual genetic profile. The HeartMap Project is a project that aims to produce a map of the human allele variants because unrelated people, so me and you for example, if we're not related, we have 99.9% of the DNA the exact same. We have the 99.9% of our DNA is the exact same, but only 0.1% contains a genetic variant that will influence how we differ as people. Analysing these genomes across 300 people across the world and mapping locations of SNPs, this will identify a set of genetic markers that can be used to screen genetic data. And these considerable genetic data is generated by genome sequencing, which is stored in biobanks. So knockout and knock-in, what is this? Well, it includes the inactivation or replacement of genes. So you can deliberately remove or add a gene which enables an organism that is concerned to function for the study of genetic disease. Gene knockout is when transgenic organisms which a gene has been removed or made inoperative. So it means you lose your gene function, which is modeled in non-human living organisms. Mice are quite used quite often, and the advantages of this is because they're biochemically and physiologically similar to a human, and they have a short life cycle. They're also easily kept in laboratory conditions. Another advantage is that they're not as ethically opposed to using mice as it would be to use larger mammals. Gene knock-in is whenever you add a particular gene rather than remove it, and this gene knock-in technology is used deliberately to add a defective gene in order to study in detail its disease progression. But obviously this may not be accurate because some animals may actually be immune to this disease and we might not be as humans, so it doesn't always work, but progress can be underpinned by ethical considerations as well. So what are the containment measures by using gene sequencing and these different things? Well, if you appropriately license and have purpose-built laboratories, then you have very effective air filters. If you have very tightly controlled procedures for staff, use special disinfectants, have bacterial strains because they're poorly equipped to survive in humans. Strains have suicide genes, so if they're activated if the conditions do not remain within a specific pH or temperature limits. And then Huntington's disease is just an example because it is caused by a single defective gene, and the condition causes the progressive brain deterioration and neurological dysfunction and death. And the worst thing about Huntington's disease is that the Huntington allele is dominant. Thank you for watching this video. I'll be Dylan Hamilton from C.A.R. Entertainment, and I'll see you next time.